Hello, I'm Professor Sims, and this video is for the Gen Bio 1, Lesson 2, Biological Chemicals and Macromolecules. This is the second of 10 lessons included in my Gen Bio 1 lecture course. In this lesson, we will define matter and elements, describe the interrelationship of protons, neutrons, and electrons. We'll explain the ways in which naturally occurring elements combine to create molecules, cells, tissues, organ systems, and organisms. We'll describe the properties of water and carbon that make them both essential for life. We'll understand the processes that make up dehydration synthesis and hydrolysis reactions. And finally, we will discuss the types and roles of carbohydrates, lipids, proteins, and nucleic acids in both animal and plant cells. The definition of matter is that it is a physical entity separate from the mind, spirit, or energy, which occupies space and possesses mass. Matter is composed of 98 naturally occurring elements. These are substances that cannot by ordinary chemical means be separated into different substances. Examples of elements are gold and carbon. Other ones would be helium, hydrogen. Elements are made up of atoms. Atoms are the smallest units of an element that retain its elemental properties. Atoms consist of protons, neutrons, and electrons. And interactions between the electrons of different atoms allow for ionic bonds, covalent bonds, hydrogen bonds, and van der Waals interactions. When two or more different types of elements combine, they form what is known as a compound. And when this happens, the elements undergo a chemical change such that they can no longer be separated once they've combined. In chemistry, a mixture is a material made up of two or more different substances which are physically combined. And the thing that makes a mixture different from a compound is that a mixture is a combination of substances in such a way that their chemical identities are retained and they can generally be separated after they've been combined. Example mixtures are solutions, suspensions, and colloids. Now, for this class, you do not need to memorize the periodic table of elements. However, you should be able to read and understand it. The periodic table shows an element's atomic mass and atomic number. The atomic number appears above the symbol for the element, and the approximate atomic mass appears below it. The atomic number is the number of protons within the nucleus of the atom. The atomic mass is an approximation, and it is approximated because it's the number of protons and neutrons in an element's atoms, or for the element's isotopes. The atomic mass is the average number of protons and neutrons existing within these isotopes. An isotope is an elemental atom with a different number of neutrons. That is, an isotope is each of two or more forms of the same element that contains equal numbers of protons but different numbers of neutrons in their nuclei. But even though they have different numbers of neutrons, they don't have different chemical properties. Protons, neutrons, and electrons are all what are known as subatomic particles. They're particles that make up an atom. Both protons and neutrons are located in the nucleus of the atom. Protons are very stable, have a positive charge, and neutrons have no charge, and electrons have a negative charge. Electrons orbit the nucleus, and there's different types of electrons, including core electrons, the ones that are on the inner shells of the electron cloud, valence electrons, the ones that are on the outer shell of the electron cloud, and those valent electrons participate in chemical bonding. Chemical reactions occur when chemical bonds between atoms are formed or broken. The substances that go into a chemical reaction are called the reactants, and the substances produced at the end of the reaction are known as the products. So this figure here is showing a reaction, a chemical reaction that is forming the product H2O, water, from the reactants, hydrogen, and oxygen. The law of conservation of matter states that matter can change form through physical and chemical changes, but through any of these changes, matter is conserved, such that the same amount of matter exists before and after the change. Matter cannot be created or destroyed, and this is important to understand when looking at chemical equations, as they must be balanced, meaning that the same number of atoms have to be present on the reactant side as there are on the product side. A homonuclear reaction is one that occurs between atoms of only one element. Homonuclear molecules may consist of various numbers of atoms, depending on the element's properties. Some elements form molecules of more than one size. This is an example. Figure 2.10 is an example of a homonuclear reaction where two atoms of oxygen combined to form an O2 oxygen molecule. Ionic bonding is the complete transfer of valence electrons between atoms. It is a type of chemical bond that generates two oppositely charged ions. In ionic bonds, the metal loses electrons to become a positively charged cation, whereas a non-metal will accept those electrons to become a negatively charged anion. Figure 2.11 is showing the formation of an ionic compound, NaCl, which is table salt. Note that the Na is losing an electron and giving it to Cl. And that is why Na is positively charged and Cl is going to be negatively charged. When atoms share electrons, they are forming covalent bonds. Covalent bonds are common in organic molecules. Organic molecules are carbon-based molecules. And they're also common in some inorganic compounds made of oxygen, hydrogen, and nitrogen. These are some examples. Carbon dioxide, it has carbon in it, so that is an organic molecule, and you can see that it has covalent bonds between the carbon and oxygen atoms. Ammonia, this is an inorganic compound, and it does have covalent bonds between the nitrogen and the 3 hydrogen atoms. And then oxygen, again, it's an inorganic compound, and it is showing covalent bonds between the 2 oxygen atoms as well. Polar covalent bonding is a type of chemical bond where a pair of electrons is unequally shared between 2 atoms. In a polar covalent bond, the electrons are not equally shared because one atom spends more time with the electrons than the other atom. And this is a function of how many protons, positively charged protons, are in the nucleus. If one atom has more protons in the nucleus, it's going to attract electrons more so than another atom that has fewer protons in the nucleus. So in that way, you can have a polar covalent bond. And these little notations here, delta negative, delta positive, is indicating that polarity. A non-covalent bond is a type of chemical bond that typically occurs between macromolecules, macromolecules being larger molecules. Non-covalent bonds do not involve sharing a pair of electrons. They are more like electrostatic interactions or hydrogen bonds. Hydrogen only having one proton in one layer of their electron cloud, so only one shell. So their bonding is rather weak. And then, of course, you have van der Waals interactions and hydrophobic interactions. So these are more like temporary attractions rather than molecules are being formed by atoms sharing atoms. They're really just kind of bumping into each other and they're more or less attracted to each other depending on their charge. In a polar covalent bond containing hydrogen, for example, an OH bond in a water molecule, the hydrogen will have a slight positive charge because the electrons are pulled more strongly towards the oxygen because the oxygen has more protons in its nucleus. Because of this slight positive charge, the hydrogen will be attracted to any neighboring negative charges. Hydrogen bonds are fairly weak and they're easy to break. And just like hydrogen bonds, your van der Waals interactions are weak attractions between atoms in close proximity. Van der Waals forces include attraction and repulsions between atoms, molecules, and surfaces. So van der Waals interactions can not only be attractions where things are attracted to each other, but they also can repel. Because remember, like charges repel, opposite charges attract. Solids that are held together by van der Waals interactions usually have a lower melting point and they're softer than those held together by stronger ionic or covalent bonds. Water is a polar molecule, like we talked about previously. Because of its polarity, each water molecule attracts other water molecules and other polar molecules. A polar substance that interacts readily with water or dissolves in water is called hydrophilic. Hydrophilic means water-loving. On the other side of it is nonpolar molecules. Nonpolar molecules like oils and fats do not interact well with water, and they're called hydrophobic. And hydrophobic literally means water-fearing. It's the observed tendency of nonpolar substances to aggregate in an aqueous solution and repel water. In liquid water, hydrogen bonds constantly form and break as the water molecules slide past each other. This phenomenon causes the water to have a high heat capacity, meaning that it takes a considerable amount of added heat to raise the temperature of water. And then as the temperature rises, the hydrogen bonds between water continually break and form again, and this allows for the overall water temperature to remain relatively stable. This is also why water has such a high heat of vaporization, which is how organisms are able to cool themselves via dew or sweat, for example. So this figure here is kind of showing that. Here you have soil that's trapping the heat from the sun, and it's heating the soil and the grass throughout the day. And then over here it's showing how the grass is able to cool itself by evaporative cooling of dew. So the dew forms during cooler nights via condensation, and then the high heat capacity of water traps heat within the dew droplets. And then as the sun heats the grass, if the grass gets to 100 degrees Celsius, then the water will evaporate and it removes that heat as it evaporates, cooling the soil and the grass to about 50 to 70 degrees Celsius. Water is a solvent, which means that it's capable of dissolving other polar molecules and ionic compounds that form hydrogen bonds with water. This results in what is called a sphere of hydration. That is, it's a three-dimensional sphere of water molecules that surround a solute. This is called a hydration shell, and this disrupts its ionic bonds and causes dissociation. Essentially, water surrounds ionic particles, breaks their bonds, and it keeps them separated from one another. Cohesion holds the hydrogen bonds between water molecules together to create surface tension. Figure 216 here is showing a sewing needle that is sort of floating on the surface of the water, and this is actually due to the fact that the surface tension of the water is pulling that needle up and suspending it. Now, since water is polar, again, it's attracted to and pulled towards other molecules. This is known as the water's adhesive force. So you can see an example of adhesion as water rises inside of a capillary tube. So we've all seen water that gets pulled up a capillary tube. That has to do with the water's adhesion. The adhesive force is where the water molecules are essentially pulled towards each other. Water can have a variable amount of free hydrogen ions. And this is due to the nature of hydrogen bonding, right? Remember, because in liquid water, the hydrogen bonds are constantly breaking and forming and breaking and forming. So the measure of this variability of hydrogen ions is referred to the pH value. That is, pH is the measure of hydrogen ion concentration in water or any other solution. A solution with more free hydrogen ions is acidic, and a solution with very few free hydrogen ions and more hydroxyl ions is basic, or also known as alkaline. And this is one of the many chemical characteristics that living things must regulate in order to maintain homeostasis. So figure 219 here is showing a pH scale goes from 0 to 14. pH of 0 is very acidic, and it has lots and lots and lots of free hydrogen ions. pH of 14 is very alkaline. It has very few free hydrogen ions. It has a lot of hydroxyl ions, the OH ions. Here are some examples of where common household items lie on the pH scale. You have lemon juice, very low pH, which means it's very acidic. You got ammonia, very high pH, which means it's very alkaline, very basic. When we talk about acids and bases, you also want to speak of buffers. Buffers can change the pH of a solution by absorbing excess hydrogen ions, which would make it more alkaline, or by absorbing excess hydroxyl ions, which would make it more acidic. So buffers aid in homeostasis and keeping the body's pH within the narrow range required for survival. For example, the buffer maintaining the pH of human blood is carbonic acid. Carbonic acid can react to form bicarbonate ions, which decreases pH and increases acidity. It can also react to form carbon dioxide in water, which will increase the pH and alkalinity of blood. So we've seen the properties of water and how it's essential to life, and now we need to look at the properties of carbon, which make it essential to life. Carbon is especially important because it does not dissolve in or react with water or acids. And with its 4 valence electrons, carbon can covalently bond to oxygen, hydrogen, and nitrogen to form the many molecules important for cellular function. Carbon has the ability to form long chains or rings of hydrocarbons, and a hydrocarbon is just the name for a compound formed with carbon and hydrogen atoms. Figure 223 is showing some examples of carbon rings or hydrocarbon rings. So we have cyclopentane, cyclohexane, benzene, and pyridine. Here is a video clip that talks more about the properties of carbon.
So what's so special about carbon? Let's look at the electrons. Carbon atoms have 6. The first 2 are in the inner shell, and the other 4 are the so-called valence electrons in the outer shell. These are the ones available for bonding with other atoms, which happens when 2 atoms share 2 electrons. Because elements in this part of the periodic table want to have 8 electrons in their outermost shell, carbon has a tendency to make 4 bonds. These can be single, double, or triple bonds, and with many different elements. In addition, while an atom that's bound to 2 or 3 other atoms will adopt a linear or flat shape, a carbon atom that's bound to 4 other atoms will take on a 3-dimensional shape, because that's how the electron clouds on the 4 atoms can be as far away from each other as possible. So it's the ability of carbon to bond in a wide variety of shapes with so many elements, including itself, that makes it so special. Also, carbon-carbon bonds are strong enough to be stable, but not so strong that they can't break and rearrange, which makes them excellent building blocks. When by itself, carbon has many allotropes, or ways an element can be arranged. There's the graphite in your pencil, which consists of slippery sheets of carbon that rub off easily. There's diamond, the hardest substance known, and we can even make interesting structures like nanotubes and fullerenes. That's just carbon by itself. When combined with hydrogen, oxygen, nitrogen, and a few other elements, it makes all the diverse structures in your body, including carbohydrates, proteins, and DNA. We can take nature a step further and synthesize novel materials with an array of purposes. Putting carbons in the empty spots of an iron lattice makes it stronger. We call this steel, which is used in most of the large structures we know today. Teflon, a carbon-fluorine polymer, is a non-stick substance that's highly resistant to chemical degradation. And carbon-based synthetic drugs like inhibitors that can silence a faulty enzyme hold the prospect of curing so many diseases. That's a lot of reasons to love carbon. It really is the element of life.
So carbon is key in forming functional groups. What's a functional group? Well, these are specific groupings of atoms within molecules that have their own characteristic properties, regardless of any other atoms present in the molecule. Common examples of functional groups are things like alcohols, amines, carbolic acids, ketones, and ethers. Figure 227 gives a nice summary of important functional groups, what their structure is, what their chemical properties are. But note that wherever you see an R, the letter R, in these chemical formulas, this R group is a stand-in for various atoms that may be attached at this location in the functional group. That is, the same functional groups can appear in many different molecules and have the same structure and function within these different molecules. Hydrogen bonds between functional groups are what helps macromolecules fold properly and maintain their shape and function. This is especially important in proteins. We're going to be talking about proteins in just a bit. These bonds are involved in recognition processes such as DNA complementary base pairing and enzyme to substrate binding as well. Figure 228 is illustrating the hydrogen bonds between functional groups of DNA nucleotides. This bonding is a key characteristic in not only the DNA structure, but also in the process of DNA replication, which is, of course, essential to life and biology. Moving on to carbohydrates. Carbohydrates are sugars, starches, and fibers like those found in fruits. They're the most abundant biomolecules on Earth. There are 3 macronutrients: carbohydrates, proteins, and fats. Carbohydrates are primarily used for structure and energy storage. They provide fuel for the nervous system and muscles. They prevent protein from being used as an energy source and enable fat metabolism. In humans, carbohydrates are important for brain function. They have influence on things like memory, mood, cognition, and even behavior. Carbohydrates are essential sugar molecules called monosaccharides, chemically linked via glycosidic bonds. Monosaccharides are classified based on the number of carbons that they have. For example, trioses, tetroses, pentoses, and hexoses. Monosaccharides are the building blocks for polymers and complex carbohydrates. So this figure here shows some examples of some monosaccharide carbohydrates. Monosaccharides, again, they're classified based on the position of the carbonyl group and the number of carbons in the backbone. So here we have aldose, ketose, triose, pentose, and hexose. Aldose is in the first carbon atom, ketose is in the second, triose is in the 1, 2, 3. Notice that the triose and aldose example are both glyceraldehyde, and that's because their structure is identical. It depends on where you count, where you start counting. Is this carbon 1 or is this carbon 1? If this is carbon 1, then you're looking at an aldose. If this is carbon 1, then 1, 2, 3, then you're looking at a triose. Ribose sugar has the oxygen joined to the 5th carbon, and hexose, glucose, oxygen is bound here to the 6th carbon. Simple carbohydrates contain just 1 or 2 sugars, such as fructose or galactose. The single sugars are called monosaccharides, but for carbs that have 2 sugars, such as sucrose, lactose, and maltose, these are called disaccharides. Then you also have polysaccharides or glycans. These are polymers composed of hundreds of monosaccharides linked together by glycosidic bonds. Energy storage polymers such as starch and glycogen are examples of polysaccharides, and they're composed of branched chains of glucose. Cellulose is a molecule that consists of hundreds and sometimes even thousands of carbon, hydrogen, and oxygen atoms. Cellulose is the main substance in the walls of plant cells, helping plants to remain stiff and upright. Incidentally, humans can't digest cellulose, but it is important in our diet as a source of fiber. Glucose storage in the form of polymers like starch or glycogen make it slightly less accessible for metabolism. However, this prevents it from leaking out of the cell or creating high osmotic pressure that could cause the cell to uptake excessive water and rupture. So in this way, starch and glycogen, again, are very important for maintaining structure and even homeostasis. Lipid molecules consist mainly of repeating units called fatty acids. Fatty acids may be saturated or unsaturated depending on the proportion of hydrogen atoms that they contain. Animals store fat as saturated fatty acids. Plants store fat as unsaturated fatty acids. Lipids are composed mainly of carbon and hydrogen, but they can also contain oxygen, nitrogen, sulfur, and phosphorus. Lipids are nutrients, carbon and energy sources, membrane structure. Lipids are nutrients. They're carbon and energy stores. They help maintain membrane structure. They act as hormones. They are used in pharmaceuticals, fragrances, and even pigments. Fatty acids are long-chain hydrocarbons with carbolic acid functional groups, and their long nonpolar hydrocarbon chains are what make them hydrophobic. Fatty acids with no double bonds are saturated, and fatty acids that do have double bonds are unsaturated. Fatty acids chemically bond to glycerol to form structurally essential lipids such as triglycerides and phospholipids. Here's a phospholipid with a hydrophobic tail and hydrophilic head. It has 2 different fatty acids. One is saturated and one is unsaturated. The unsaturated fatty acid has a slight kink in its structure, which kind of makes it look like it's bent, like a bent leg. And this is because of the double bond. Triglycerides, like the name suggests, are made up of 3 fatty acids bound to a glycerol. Triglycerides are hydrophobic, like the hydrophobic hydrocarbon chains of phospholipids. Biological membranes are made from 2 layers of phospholipids with a hydrophobic or water-hating interior and a hydrophilic or water-loving exterior. Steroids are lipids with complex ring structures that function as structural components of cell membranes and also as hormones. Cholesterol is a type of steroid, and it's part of the plasma membrane, but in addition to that, cholesterol is a precursor of the steroid hormones such as testosterone. Phospholipids tend to arrange themselves in aqueous solution, forming liposomes, micelles, or lipid bilayer sheets. So again, the hydrophilic polar head group and the hydrophobic tails are depicted here. A protein is any of a class of nitrogenous organic compounds that consists of large molecules composed of one or more long chains of amino acids. Proteins are an essential part of all living organisms, especially as structural components of body tissues and as enzymes and antibodies. In proteins, the structure determines its function. Protein structure is very important, and it is classified as primary, secondary, tertiary, or quaternary. If a protein's native structure is disrupted, for example by denaturation, then its biological function is lost. Here is a video clip that's further describing the nature of proteins and the importance of their structures.
Proteins are synthesized by polymerizing amino acids. The polymerization occurs by repeatedly forming peptide bonds that link individual amino acids together into a chain. There are three structural features that influence the three-dimensional shape of a water-soluble protein. Primary structure is the peptide bond between individual amino acids that creates a long chain of connected amino acids. These long chains of polymerized amino acids have hydrophobic, water-repelling, and hydrophilic, water-attracting projections that are oriented perpendicular to the chain, as shown in this illustration of a growing protein chain. Secondary structure is the helix that the protein chain curls into as a result of hydrogen bonds and other weak forces. Tertiary structure is created when the protein molecules fold back on themselves outside of the helical segments to put the hydrophobic portions to the interior and the hydrophilic portions to the exterior. Several helical regions can exist in different portions of the molecule. When the protein has folded and refolded to reach its most stable configuration, it will have mostly hydrophilic amino acid residues on the exterior and mostly hydrophobic residues directed into the interior. When natural proteins are subjected to physical or chemical treatment, their structure changes and they become unnative or unnatural. We call that process denaturation. In this example, heating the proteins in solution imparts energy to the protein molecules. This added energy is enough to break the relatively weak forces that hold the protein in its refolded and helical tertiary and secondary configurations. As the process of denaturation proceeds, the protein molecule unfolds more and more, and the internally directed hydrophobic regions now become exposed on the outside of the molecule. The peptide bonds are largely hydrophilic. Once these segments are set free from each other, they attract water molecules. The recruitment of water molecules entraps the water molecules in close proximity to the protein strands. The hydrophobic portions of the molecules are also exposed. This situation is unfavored because the hydrophobic portions of molecules are not stable in an aqueous environment. Hence, upon unfolding, the hydrophobic regions on individual protein molecules will associate with hydrophobic regions on other protein molecules. This situation encourages the association of these protein molecules into larger and larger random three-dimensional structures. The molecules aggregate into very large water-insoluble collections that are quite randomly assembled. As the proteins denature, latticework structures grow amorphously and attract the solvent water molecules into cell-like structures. The self-associated water molecules, in groups, adhere to the surface of hydrophilic regions of the protein, while hydrophobic regions of the protein dissolve into each other and provide the energy to retain the structure. As this process continues irreversibly, all of the protein molecules are recruited to this large insoluble mass in a randomly organized structural framework that contains entrapped water molecules. One example of the consequences of unfolding and reassociating protein molecules is coagulation of egg white. Frying an egg is no more complicated than denaturing the egg white protein.
Figure 3.24 is showing the schematic diagram of peptide bonds that link amino acids together. These bonds are formed via dehydration synthesis reactions that link the carboxyl group of one amino acid to the amino group of another amino acid. This reaction is termed dehydration synthesis, because when these two link together, a hydroxyl and a hydrogen are released, and then those two subsequently form a water molecule. Here is a schematic diagram of the primary structure of a protein. It is simply a long sequence of amino acids that are linked together. Finally, this is a summary of the 4 levels of protein structure. Here's the primary structure that you just saw, the sequence of amino acids. Secondary structure is when the primary structure has begun folding, and it exists either in sheets or in helices. Tertiary protein structure is 3-dimensional folding pattern, and the main difference between secondary and tertiary is because tertiary is involving side chain interactions at this point. So it's not just folded in on itself. It's like 3-dimensional folding. And then the quaternary structure is when you have more than one primary structure that's folded together. So it's more than one chain of amino acids. Now we have nucleic acids. Nucleic acids are the most important macromolecules of biology and indeed of life because they carry genetic blueprints for functioning and replication of cells. They carry the DNA and the RNA. We have a table here that lists the features of DNA and RNA. It lists their function, location, structure, the type of sugar that it contains, and in which purines and pyrimidines they contain as well. So you have DNA. DNA carries genetic information. It is in the nucleus. Its structure is a double helix. The sugar is deoxyribose. And then you have the cytosine, thymine, adenine, and guanine as their nucleic acids. It also carries genetic information, but it's primarily involved in protein synthesis. It does leave the nucleus via the mRNA, the messenger RNA. RNA is single-stranded. Its sugar is the ribose, and it has cytosine, adenine, and guanine, just like DNA, but instead of thymine, RNA is going to have uracil. Nucleic acids are composed of nucleotides, each of which contains a pentose sugar, a phosphate group, and a nitrogenous base. So it has to have all 3 of those things: a sugar, a phosphate, and a nitrogenous base. Nucleotides are linked together by phosphodiester bonds between the 5' phosphate group of one nucleotide and the 3' hydroxyl group of another. So the 5' and the 3', that just is, like we saw earlier, it's just the number of the carbon that's in the ring. Okay, so when it says the 5' phosphate group, that's the phosphate group that has bound to the 5th carbon, right? And then the 3' hydroxyl group is the hydroxyl group that has bound to the 3rd carbon. So that's what those numbers mean. DNA has a double helical structure with 2 anti-parallel strands of deoxyribonucleotides connected in the middle by hydrogen bonds. RNA is single-stranded, and it is composed of nucleotides made up of pentose sugar, which is ribose, and nitrogenous base, and a phosphate group. The structure and orientation of DNA is essential for its replication. So the DNA strands are both complementary and directional, and then during replication, each strand copies itself, resulting in a daughter helix containing 1 parental strand and 1 newly synthesized DNA strand. We are going to discuss this in much more detail in later lessons. For now, let's go to RNA and its subunits. These are, again, mostly involved in protein synthesis. This is a process called translation, and I'm going to show you a short video of what that looks like now.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a 3rd site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the 2nd amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics, and leave your questions for me in the comments below.